Scientists would never have become fascinated with strawberries if it wasn't for this plant pigment. Fisetin is a promising anti-aging compound, a so-called senolytic. It has anti-inflammatory properties, antioxidant and immune supporting effects. It's a flavonol, a yellow pigment that belongs to the flavonoid group of polyphenols. It gives colour to many different fruits and vegetables. Compared to now famous plant antioxidants like resveratrol and quercetin, Fisetin was unfairly ignored for too long. It wasn't until recent years that research became increasingly interested in its medicinal potential. Science teams are currently exploring its ability to slow the aging process and to extend lifespan, its so-called senolytic effects. And what's more, the Fisetin has powerful anti-inflammatory, antioxidant and immune supporting properties. Despite promising animal and cell-based finding, research is still in the early stages. Only a single clinical trial has been conducted with Fisetin. It may protect the brain and improve memory, delay aging, help to control blood sugar, have anti-cancer properties, and protect the liver and the heart. Many fruits and veg contain Fisetin. The highest concentration is found in strawberries, apples, persimmons, lotus root, onions, and grapes. Fisetin increases the antioxidant defense. It directly neutralizes free radicals, and it also increases other powerful antioxidants such as glutathione, superoxide dismutase, and catalase. Secondly, it can block a pathway called NF-kappa B. This is a switch that tells the genes to produce inflammatory compounds. An overactive NF-kappa B response is linked to allergies, autoimmune disease, and cancer. And Fisetin blocks inflammatory enzymes that degrade fatty acids. It also reduces the activity of a group of enzymes that cancer requires to spread and to invade to other tissues. mTOR's demands for energy and growth control your cells into a frenzy. Metabolic waste builds up, and there's no time to clean it out. An overactive mTOR response is associated with cancer, diabetes, obesity, and neurodegenerative diseases. By blocking this pathway, Fisetin helps to remove waste and to enhance cellular metabolism. Blocking the mTOR increases longevity. In fact, blocking the mTOR is the only intervention that increased lifespan in all organisms studied to date. This is because once the mTOR is blocked, autophagy is activated, a process of recycling damaged cellular components. Much of the interest in Fisetin is centered around its potential to help to slow aging and to extend a healthy lifespan. Aging is characterized by the buildup of senescent cells. Cells that stop dividing, become damaged, and start releasing inflammatory molecules. As we accumulate more and more of the senescent cells, the body starts being affected. These cells start damaging the healthy tissue. They contribute to many age-related diseases, from osteoporosis to cancer to heart and brain disease. Removing senescent cells calms the inflammation. It improves physical function and increases lifespan in animals. Certain plant compounds are able to destroy senescent cells without harming the healthy ones. In a 2018 cell-based study of 10 compounds, Fisetin was the most effective one. In old mice, Fisetin cleared senescent cells and it increased their lifespan by over 10%. Because of this study, researchers have become particularly interested in the senolytic or senotherapeutic properties of the Fisetin. It also extended the lifespan of yeast by more than 50% and the lifespan of fruit flies by more than 20%. Because of these promising results, a clinical trial is underway to see if the Fisetin is effective for reducing inflammation and improving frailty and bone health in the elderly. In multiple animal studies, Fisetin restored blood sugar levels of diabetic rats and mice to those of healthy animals. It improved their ability to control blood sugar levels by increasing insulin levels, increasing enzymes that turn sugar into energy, removing sugar from the blood to store as glycogen in the liver, and reducing the liver's ability to make sugar from lactate and amino acids. Fisetin slowed the progression of cataracts, and it protected the kidneys of diabetic mice by blocking inflammation and oxidative stress. It also protected the liver of diabetic rats from high blood sugar levels by increasing antioxidant levels. Another complication of diabetes is the hardening of the arteries and heart disease. In a cell study, Fisetin prevented high sugar levels from causing inflammation in blood vessels. Inflammation is linked to colon cancer growth, as well as its spread and resistance to chemotherapy. In a clinical study of 37 colon cancer patients undergoing chemotherapy, the Fisetin reduced the markers of inflammation. However, the authors didn't report the effects on tumor growth and progression. In rats, Fisetin reduced oxidative stress and growth of liver cancer caused by fungal toxins. In mice, it prevented the growth of lung cancer, and it boosted low antioxidant levels caused by a toxin in tobacco smoke. It reduced lung tumor growth by 67% in mice, and by 92% when combined with a chemotherapy drug. It also prevented the growth of new blood vessels, supplying nutrients to the cancer. Another study found that Fisetin reduced tumor growth by 66% in mice with melanoma. Certain types of prostate cancer are fueled by androgens like testosterone and dihydrotestosterone. Fisetin slowed the growth of prostate tumors in mice by blocking the receptors for testosterone and DHT on the cancer cells. Fisetin also protected against kidney damage from chemotherapy in rats by reducing inflammation and boosting antioxidant levels. In cell studies, Fisetin caused programmed cell death, and it prevents the growth and the spread of a variety of cancer cell lines, but this doesn't imply the actual anti-cancer effect in living organisms. Older rats given Fisetin experienced memory and learning improvements. Cell studies revealed that Fisetin activates pathways in the brain that are involved in storing memories. In another study, Fisetin prevented memory loss in mice who were exposed to toxins. Fisetin can easily cross the blood-brain barrier in mice. This is important as there is an ongoing debate about whether compounds like Fisetin can reach high enough levels in the brain to improve its function. Fisetin reduced depression and anxiety in mice by increasing levels of serotonin and noradrenaline, neurotransmitters that play key roles in mood. Immune cells in the brain, called microglia, are overactivated in neurodegenerative diseases like Alzheimer's disease and Huntington's disease. This causes inflammation and damage to healthy brain cells. In cell studies, Fisetin boosted brain antioxidant levels, and it prevented microglia from releasing inflammatory compounds in response to bacterial toxins. Alzheimer's disease involves the buildup of amyloid plaques and tau protein in the brain. Fisetin reduced the level of tau protein in the brain by activating a process that removes these harmful proteins called autophagy. In mice with amyloid plaques, Fisetin improved memory, reduced inflammation, and prevented the loss of brain cell function. In mice with Alzheimer's, it reduced amyloid plaque buildup and loss of brain cells. Huntington's disease is a genetic movement disorder where the brain cell gets destroyed. In mice with Huntington's, Fisetin was able to improve declining physical function and increase lifespan. In amyotrophic lateral sclerosis, or ALS, brain cells that control the muscles die off. Fisetin improved the balance and the muscle coordination, and it increased the survival in the mice with ALS. Fisetin protects brain cells, and it reduces inflammation and damage due to stroke in mice and rats. In another study of rabbits, Fisetin prevented loss of balance, lack of energy, and uncontrolled eye movement caused by stroke. Cell studies show that Fisetin reduces the activity of immune cells in the brain that are responsible for the inflammation and brain damage after a stroke. Aluminium is toxic to the brain, and it's been linked to Alzheimer's disease. In mice, Fisetin reduced inflammation and oxidative stress in the brain caused by aluminium. In cells, Fisetin promotes the survival of brain cells by getting rid of damaged and unneeded proteins. In a mouse model of brain trauma, Fisetin prevented seizures by reducing oxidative stress. Fisetin also reduced seizures and death in mice who were exposed to chemicals and electric shocks. It did so by increasing the levels of the neurotransmitter GABA and reducing oxidative damage in the brain. By reducing oxidative stress and inflammation, Fisetin protects the brain and it improves cognition and mental health. Fisetin reduced high total and LDL cholesterol and triglycerides in rats who were fed a high-fat diet. In diabetic rats, it doubled the HDL levels and it cut LDL cholesterol levels in half. A cell study hinted that Fisetin reduces cholesterol by causing more of it to be released in the bile. Cell studies show that Fisetin prevented immune cells called macrophages from oxidizing and ingesting the LDL cholesterol. When macrophages ingest oxidized LDL, they create fatty plaques that harden the arteries and cause heart disease. In rats, Fisetin improved the poor blood flow caused by a high-fat diet. Test tube experiments reveal the Fisetin helps to relax and dilate blood vessels, and this helps to improve blood flow and reduce blood pressure. Fisetin protected heart cells from oxidative stress, and it improved heart function in rats with abnormal thickening of the walls of the heart. It also protected heart tissue and mitochondria function from damage due to a heart attack in rats. According to animal and cell studies, Fisetin can support the heart health by reducing high cholesterol levels, improving circulation, and protecting the heart from oxidative stress. In mice, Fisetin protected the liver from alcohol by helping the animals to process it quicker. It also reduced oxidative stress, which prevents damage. In mice and rats who were fed high-fat diets, Fisetin reduced the buildup of fats in the liver. It worked by increasing enzymes that break down the fats and reducing enzymes that make new fats. Diabetes can often cause nerve damage and pain. Fisetin reduces the heightened sensitivity to pain in diabetic mice and in mice with nerve injuries. It lowered oxidative stress and it increased serotonin and GABA activity in spinal nerves. This acts to relieve the sensation of pain. Estrogen keeps bones healthy. After the menopause, low estrogen levels put women at risk of osteoporosis. The rise in inflammation due to aging also weakens the bones. Fisetin improved bone density, and it prevented bone loss in mice with low estrogen levels and inflammation. In cells, it worked by reducing the activity of bone-degrading cells called osteoclasts. Collagen gives the structure and the elasticity to the skin. In human skin cells, Fisetin prevented the breakdown of collagen from UV sun exposure, a key factor in skin aging. It also reduced inflammation and oxidative stress caused by UV rays. Applied to the skin of mice, Fisetin prevented the abnormal growth of skin cells, DNA damage and inflammation caused by UVB rays. It also reduces the formation of wrinkles by boosting skin collagen. Skin inflammation and eczema is typically treated with steroid creams, which often have harsh side effects. In one study, it reduced skin inflammation, swelling, and redness in mice with eczema. Fisetin has the potential to protect the skin from excessive UV exposure, which can cause skin damage and aging and relieve eczema. In one study, Fisetin helped to prevent listeria infection by interfering with the bacteria's ability to hide from the immune system. In another cell study, it was active against two fungi that cause infections in people with weak immune systems. It impairs the production of a compound that fungi need to survive, called ergosterol. Fisetin can also help with other infections caused by parasites. It blocks the activity of arginase, an enzyme that parasites need to protect themselves. Immunoglobulin E antibodies and T immune cells activate mast cells and basophils. This goes on to trigger an allergic response. In cell studies, the Fisetin prevented the T cells and the immunoglobulin E from activating these cells and causing inflammation. Similarly, Fisetin prevented immunoglobulin E antibodies from activating the mast cells in mice who were exposed to allergens. In turn, it stopped the release of histamine and other inflammatory compounds. In a mouse model of inflammatory bowel disease, or IBD, Fisetin reduced damage and inflammation in the colon by blocking NF-kappa B activity and restoring antioxidant levels. Even at high doses, scientists found no evidence of side effects or toxicity in animal studies. Clinical studies, of course, are needed to confirm its safety. In a lone clinical trial on cancer patients, stomach discomfort was reported in the Fisetin group. However, this might not actually be a side effect of the Fisetin. All patients were also receiving chemotherapy at the same time that the stomach complaint was also reported in the placebo group. Due to the lack of safety data, pregnant women and children should avoid using Fisetin. Supplement-drug interactions can be dangerous and in rare cases even life-threatening. Always consult a doctor before supplementing and let them know about all the drugs and supplements that you're using. The liver uses the same pathway to process Fisetin as it does for the blood thinning agent warfarin. This could in theory increase the effects of warfarin. Fisetin substantially reduces blood sugar in diabetic animals. The combination with blood sugar lowering drugs may further reduce the sugar levels. In a clinical study of colon cancer patients, 100 mg per day was effective for reducing inflammation. In ongoing clinical trials looking at the effects of Fisetin on inflammation, bone health and frailty in the elderly, Fisetin would be used as high as 20 mg per kg for 2 consecutive days. This would be around 1,400 mg per day for a 155-pound person. There's at least one simple way to theoretically increase its absorption. Take it with fats, as Fisetin is fat-soluble, similar to other flavonoids like quercetin. Fish oil and other oils enhance the bioavailability of quercetin, and they could do the same for Fisetin. Even so, new formulations combining Fisetin into small fat-like molecules called liposomes may be the only effective solution. These greatly improve its absorption and anti-cancer effects. For more information about herbs, supplements, and natural treatments, check out my website.